Greetings everyone, and today we are going to discuss about virus. Previously we are done with our chapter which is bioenergetics, and now virus includes first its most basic introduction. What is virus? Virus is neither a living organism nor a non-living organism. In our previous classes, we have studied that it is an intermediate organism between the living and non-living. It is an intermediate entity, and now we are going to discuss that this is an infectious particle. It is an infectious particle, as well as it is the smallest entity, smallest particle, which is considered uh, in smallest entity which is considered in biological sciences. And here, this smallest particle is basically having no cellular particles. It is not having a cellular composition. No cellular organelles are there. No nucleus. Nothing is there. Instead of this, there is presence of some genetic material, some genetic material which sustains it, which is known as genome, as well as there is the presence of an outer protein coat which is known as capsid. Protein coat known as capsid. Now that protein coat is known as capsid. It's about the general structure, but in some viruses we are also having some other general characteristics which we are going to discuss in our next slide. Here, as you can see, there's an example given on the screens that here there's a virus which is quite smaller in size. When generally talking about size of virus range in size from 10 nanometer to 300 nanometer. Moreover, these are having characteristics. Some good MCQs that largest virus is a pox virus, or we can also call it as smallpox virus. By the way, pox virus is also divided into several kinds, such as um, vaccinia virus, vaccinia, cowpox virus. It is the largest smallpox virus, which is in size around 500 nanometer. Its size is similar to mycoplasma. Some viruses are having their size similar to mycoplasma bacteria, mycoplasma, which is not having a cell wall. So, mycoplasma bacteria are so small that they are having their size resembling to those of viruses. Uh, Not all viruses, but the larger viruses. Vaccinia, cowpox, as well as variola, smallpox virus, it's around 250 nanometer in size. Now coming to some other parts, viruses do not have their own metabolic machinery. What does this mean? This means that viruses do not have the capability to synthesize the material required for their metabolic needs by their own self. So they depend on the host's metabolic machinery in order to synthesize the material they need in order to sustain themselves in this world. So um, example of this is bacteriophage. Now when we are going to discuss about classification will also classify m- the viruses on several classes, but here we are going to just discuss about generally about a virus which is known as bacteriophage. It is a virus which can attack bacteria. Virus which attacks bacteria is known as bacteriophage. Viruses attack all types of organisms from bacteria to human beings. So here, um, bacteriophage Bacteriophage, for suppose, is a virus which is quite specific and only attacks the bacteria. And once it enters inside the bacterial cell, it hijacks the central dogma of the bacterial cell. As it hijacks the central dogma, it tends to create protein of its own choice, as we have previously discussed in some of our classes as well. As it enters inside the bacterial cell, it causes to utilize the bacterial metabolic machinery, uses the bacterial ribosomes in order to synthesize its own material. Now, uh, based of these functions, uh, some of the viruses are found in different shapes, different sizes, as well as uh, some common characteristics of virus which make them uh, come on the borderline between living and non-living is such that, first let me just write it down so that we can thoroughly discuss each and everything. Living, non-living. characteristics. When it comes to living characteristic, its living characteristic is such that it can replicate. It can replicate. It can replicate itself, reproduce. While one more thing, that reproduction is non-vital for living organism. Living organisms can survive without reproduction, but virus cannot survive without reproduction because it is a vital process for virus. This is the only reason the virus tends to go inside the host cell, infect the host cell, make its multiple copies. Once it makes its multiple copies, it forms um, uh, multiple copies, as a result of which it causes the cell to become infected and kill the cell ultimately. Non-living cells are now here a living characteristic that it can get replicated. It has genetic material, genome. It contains genome, as well as it is having characteristic that it can get damaged by change in enzymatic, change in pH, change in pH, temperature, as well as change in pH, temperature, change in get damaged by change in pH, temperature, and it can also be damaged by ultraviolet rays. As our DNA, as living organisms can have their DNA mutated by ultraviolet rays, their genome can also be mutated by ultraviolet rays. Moreover, moreover, here we are also having some other characteristics in non-living, that here non-living characteristics, they do not have their metabolic machinery, don't have metabolic machinery. Moreover, they basically, they do not have metabolic machinery. Oh, they do not have metabolic machinery, as well as they do not have cellular organelles, as well as outside the cells. They are non-living outside the cells, which means they cannot multiply themselves outside the cell, which is a non-living characteristic. Outside the cells, can't multiply. Can't multiply outside the cells. Can't multiply outside the cells, as well as they are non-living in vitro, and they can also be crystallized. They can be crystallized, which indicates that these are non-living. And they don't perform digestion, excretion, and respiration. So these are some differences. Now when coming to our next part, which is about the general structure. General structure of virus can be differentiated in such a manner that here, as you can see, a virus. Basically, a virus is made up of two major parts. First major part includes the genetic material genome, which is also known as the inner core, or in simple words, we can also call it as a core. And the outer part is known as the covering part. This covering part is known as the protein coat. The outer part is known as the covering part, which is known as a protein coat, known as capsid. The virus collectively can be called as a nucleocapsid or nucleoprotein. Why it can be called as nucleoprotein? Because it is made up of nucleoprotein and nucleic acid and capsid. So it can be named as nucleocapsid. Moreover, moreover, in some viruses there is also a presence of lipid bilayer. These viruses which are having lipid bilayer as an envelope, as an envelope, and these viruses which are having lipid bilayer as an envelope are known as enveloped viruses. Enveloped viruses. These are known as enveloped viruses. And example of these includes HIV, hepatitis, hepatitis B are enveloped viruses. Um, Rhinovirus is an unenveloped viruses, non-enveloped viruses. Rhinovirus is a non-enveloped virus. When we come to describe their mode of because they behave as parasites, so these can be considered as their mode of living can be considered as parasitism. So we can consider them as obligate intracellular parasite. What does this mean? This means that these are obligate, which means they cannot live without their host cell. Intracellular, these are the only parasites which are intracellular, and parasites because they harm their cell and harm the host cell and get benefit from it. Now these were a few things I wanted to discuss over here. Now while uh, when it comes to the lipid bilayer enveloped viruses basically are also having a characteristic that these are having a coating fusion of lipid bilayer, which is derivated lipid bilayer, as well as presence of glycoprotein is also there over their structure. Glycoprotein spikes are present over their structure. Glycoprotein spikes are present over their structure. Spikes are present over their structure, which are utilized for cell recognition because viruses need to enter the host cell by first adhering itself to the surface of the host cell, as a result of which it requires a receptor in order to adhere itself to the cell surface membrane. So in that case it has glycoprotein spikes. I'm talking about the envelope virus. It has glycoprotein spikes which are utilized over here. One more thing over here, that these glycoprotein spikes can vary depending on the type of virus it is. For example, if we are having HIV, HIV is having glycoprotein spikes known as GP41 and GP120. Moreover, uh, when it comes to non-enveloped viruses are basically having some other structures for other kind of spikes for recognition of the host cell, which are known as polymers. Polymers. Yes, these are known as polymers. Now, these polymers are again made up of capsomere subunits. What are these? We are going to discuss right now. That capsid. Capsid is basically it is a protein coat. That protein coat is made up of several um, protein subunits known as capsomere. Now, capsomere basically it is a protein subunit, capsomere, which is having different structures. Um, Let us generally discuss about tobacco mosaic virus (TMV). Tobacco mosaic virus basically is having 2,130 capsomeres, and its structure 2,130 capsomeres in its capsid, which are arranged helically, which are arranged helically, which are oops, which are arranged helically. As a result, it gives a road shape. They are arranged helically, to, due to which the shape which it gives to the virus is a hollow coil shape. Hollow coil shape or road shape. Hollow coil shape or road shape. Now, each of the capsomere present within the tobacco mosaic virus is made up of 158 amino acids. There are other examples as well. For suppose if we are having a herpes virus, herpes herpes virus is having 162 capsomeres, while adenovirus, both of these viruses are basically non-enveloped viruses. Adenovirus, these are 252 capsomere containing viruses. Capsomere containing viruses. 252 capsomere containing viruses. Moreover, both are non-enveloped viruses and both are having polyhedral shape. As they are different shapes, um, herpes and adenovirus are polyhedrons, whereas tobacco virus, mosaic virus is having a road shape because of the helical structure. Moreover, when it comes to its further detail, we are going to discuss it in the classification part. One more thing, their genome. The genome basically involves uh, two categories, two major categories. They either have a DNA or have a RNA. They do not have DNA or RNA together. They can have either a single strand of RNA, or they can have one strand of RNA or two strands of RNA, or one strand of DNA, one two strand of DNA. Now here, DNA can be single stranded DNA, or it can be a double stranded DNA. Likewise, RNA can be single stranded RNA, or it can be double stranded RNA. Likewise, the single stranded RNA is further classified into plus stranded RNA, plus stranded RNA virus, or positive sense single stranded RNA virus. Likewise, there is minus stranded RNA virus as a type of single stranded RNA virus. Basically, a minus stranded RNA virus can also be called as negative sense single stranded RNA virus. Moreover, there are other types as well, such as retrovirus. Retrovirus are basically those viruses which perform reverse transcription. Those viruses are known as retroviruses which perform reverse transcription. These are having special enzyme. These are having three enzymes particularly, which are known as reverse transcriptase, reverse transcriptase, integrase, protease. These are three enzymes containing. Now, retroviruses are further classified into their types, which includes are single-stranded RNA virus with DNA intermediate, double-stranded DNA virus, double-stranded DNA virus with RNA intermediate, RNA intermediate. So these are some common classification. We are going to further discuss that in detail in the next slide. uh, One more thing that here, uh, there are different shapes of viruses. First includes a polyhedron shape. There are two major classes of shapes as well. First includes a polyhedral shape, and the second one includes the helical shape. These two words refer to different dimensions. Here, polyhedral means 3D shape, helical means 2D shape. So here, helical shape can be divided into road shape either. Road shape, example is TMV, or circular shape. Circular shape, example is HIV. Polyhedral shape. Polyhedral shape is a shape which is having polyhedral. Basically, it's not the complete part. It is icosahedral. It is icosahedral. Icosahedral means a shape which is having 20 equilateral triangles or 20 faces. 20 equilateral triangles, 20 equilateral triangles or faces. Now, how is its structures formed? Basically, these triangles are arranged in 3D in such a manner that they are having 20 faces, 12 vertices. Moreover, icosahedral basically now is divided into its classes, into its types, which includes polyhedral shape, and spherical shape. Now you might be wondering how spherical shape can exist in a icosahedral, because icosahedral are in triangle shape. So we have studied somewhere in maths, in geometry, that if there is an equilateral triangle and that is present within a circle, that can uh, have a common center with the circle, because all of the sides of the equilateral triangle touch the circumference of the circle. At that moment, the center point is known as circumcenter, which you might have studied, and with the consequence of this point, the spherical shape can be obtained. Moreover, when it comes to some other parts, for example, if we are going for the polyhedral shape, basically within this structure of polyhedron and spherical shape, the 12 vertices and 20 faces are having several capsomeres. 12 vertices are having pentagonal capsomeres. 12 vertices are having pentagonal capsomeres known as pentones. These are constant in all of the polyhedral and spherical viruses. While there are 20 faces, on the 20 faces, there are around hexagonal. There are hexagonal capsomeres. Hexagonal capsomeres, which are known as hexons. These hexons basically are variable, vary from virus to virus, but the 12 vertices are having 12 pentones, which are common. Now, um, some common one more thing, poliovirus. Poliovirus, picornavirus is having 32 capsomeres. All right. Now, these are the classification: road shape, circular shape, polyhedron, and spherical shape. One more classification is there, which is a complex classification, which is the bacteriophages. Bacteriophages are those which attack bacteria. These are having a complex structure, which means these are having complex shape. Their structure is divided into three parts, which is includes the head, neck, and tail. As you can see in the diagram, uh, let me just zoom it a bit. Yeah. So as you can see in the diagram, this diagram showcases the bacteriophage, which is having um, a tail part and a head part. Head part is polyhedron, tail part is rod shape. And this structure in which polyhedron is head shape and rod shape is the tail part one, it is known as a complex structure. It is classified in its classification, which is based on upcoming topics. Now coming to the discovery, its discovery is basically based on a number of things. First and foremost, in early age, there was an assistant of Louis Pasteur known as Charles Chamberlain. Charles Chamberlain invented a invented a filter known as porcelain filter, a porcelain water filter, or we can also call it as Chamberlain Pasteur filter. He basically utilized this filter in order to check that what kind of infectious particles can be passed through, can be you know can be filtered out. How how the infectious particles of the tobacco mosaic virus can be filtered out using the porcelain filter paper. According to our book, it is having the characteristic, it is having the point that the porcelain filter paper can only allow to filter particles which are either equal to or greater than zero point 0.2 nanometer. Their diameter of their pore is equal to or greater than zero point 0.2 nanometer. As a result of which, it has basically it can allow all type of bacteria to be filtered out over the filter paper, but cannot cause the other particles. For example, if we are having viruses were not filtered out using that. So he was the person who invented it. Now, after the Dmitry Ivanovsky, by the way, he also gave the name virus, which is derived from the Latin word venom, meaning poisonous fruit, slimy fluid, or water fluid. In simple words, we can also call it as venom or poison. According to me, it is Latin word. Moreover, if we come to Ivanovsky, when we come to Ivanovsky basically when he actually did an experiment, that when he actually took this a pulp of infected tobacco mosaic plant and passed that infected pulp through the porcelain filter paper. When he passed it through the porcelain filter paper, he he already knew, because at that time it was quite common that all were not known about viruses, and they thought that tobacco mosaic disease is caused by a bacteria. So when he passed through them, all of the bacteria were filtered out. But after the bacteria were filtered out, the rest of the material which was left behind, the filtrate which was left behind, was applied over healthy tobacco plants. As a result of which, they were also infected. At that point, he found out that there is an other infectious particle smaller than bacteria, which is infecting the tobacco plants, not a bacteria, but a particle smaller than it. And one more thing, bacterias are 10 to 1000 times larger, 10 to 1000 times larger than viruses. And according to our book, according to our book, Now we are going to discuss about our next part. When a new scientist came, whose name was Bejenrak. M W Bejnirak. Bejnirak, something like that. I'm not sure about the real spell. You can search it out on the Google. Bejnirek. So Bejnirek basically did the same experiment which was conducted by Ivanovsky, but that time when he took the filtrate, he uh, applied the filtrate over the healthy leaves, due to which the healthy leaves were infected. And when he utilized those healthy leaves which were infected, he came to knew that this pathogenic fluid, this filterable pathogenic fluid, this filterable pathogenic fluid was named by him as contagion, 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 vivicum, contagion, vivum, contagion, vivum fluid, also known as contagious living fluid. Later, it was named as virus by Mr. Ivanovsky. Now, in 1935, W. N. Stanley was a scientist who came. W. N. Stanley, who performed the crystallization of virus. Now, what he did was that he actually utilized the method of crystallography, in which he crystallized viruses in solid form. Now, in that case, this also indicated that virus has characteristics of non-living as well as living. After that, electron microscope was discovered. Further chemical characteristics of viruses were discovered, and then it was found about its general structure, its chemical composition, including its um, chemical structure of capsid as well as genome. Coming to next part, which is its classification based on geometry. Classification based on geometry is clear, which I have taught you right now. Classification based on host in such a manner that they are classified on the basis of their host. For example, zoophages, phytophages, bacteriophages, mycophages, phycophages. So coming to phytophages first. Phytophages are those viruses which infect plant. There are more than 2000. These are mostly RNA viruses. The viruses which infect plants are mostly RNA viruses. Some of these are DNA as well. And zoophages are those which infect animals. These are mostly half of these are DNA, half of these are RNA viruses. Bacteriophages are DNA viruses, and mycophages and phycophages are viruses. Mycophages are uh, those viruses which attack fungi, and phycophages are those viruses which attack algae. Now, phytophages basically involve, there are more than 2000, there are more than 2000 RNA viruses in phytophages. Examples of these includes the tobacco mosaic virus, um, CLCV. Basically, CLCV is a spherical virus. CLCV is a spherical virus, as well as when it comes to CLCV and CAMV, cauliflower mosaic virus and cotton leaf curl virus. Both of these are viruses which are basically um, these cauliflower mosaic virus and cotton leaf curl virus are the two viruses which are DNA viruses. Both of these are single-stranded DNA viruses. One more thing, that in phytophages are never double-stranded DNA viruses. Double-stranded DNA viruses are only found in humans and other types of in animals as well as other types of organisms, but not in plants. They are only single-stranded DNA viruses. Single-stranded DNA viruses are single-stranded DNA viruses. Single-stranded DNA viruses.
इन दोनों में स्पेस देने के लिए होता है।
Here, as we have
मार्जिन का जो
discussed
टैग
about
है,
more than
जो
2000
मार्जिन हम
RNA
लगाते हैं, वो
viruses like
मार्जिन आप
CMV,
देख के जो
CLCV,
सामने खोलते हैं,
CAMV,
और पेंटिंग भी यही
common
काम
ones.
करती है, उसकी
There are
जो
other
साइज़
viruses
है
as well,
बड़ा छोटा-सा नए-नए
as well
पेंटिंग
as
तब आती है,
there are other
इसलिए मैंने
viruses.
एक
Example of this is
लिंक भेजी
lepto
थी, इंस्टाग्राम की रील भेजी थी
spindle,
मैंने क्लब में, आज मैं दोबारा भेज दूँगा, मार्जिन और पेंटिंग में डिफरेंस क्या है, ठीक है?
potento
उसके बाद
spindle bar
टैच
virus,
डेकोरेशन के बारे
TSPV,
में, जैसे
potato
डेकोरेशन करना
spindle
हो तो
mosaic
फिलहाल
virus, sorry,
नेपाल में तो कोई ऐसा टैच डेकोरेशन
TSMV,
होता नहीं है, जो हमने वो
mosaic
नन कर दिया।
virus.
और
Uh,
दैट्स
but
एट
another
हाउएवर मतलब जैसे मैं
country,
इसमें देख
top
क्लिक कर रहा
virus,
हूँ, इसका
they
कलर
have
देखो
like
ग्रे
many.
आ रहा है, तो ये हमने उसमें लगाया है कि
One
जैसे
more
हम
thing that
क्लिक करें तो
here
उसका कलर ग्रे
water
आना
leaf curl
चाहिए।
virus is
और
carried
फिर
by,
इसके बाद जो
it
हमने
is
नीचे सर्च मिल रही है,
infected, it
जो
is
हमने
taken by a
सर्च
carrier.
वाला
There are
ये
some
ऑप्शन
viruses which
है,
are also
इसकी
infected
हमने
by
सीएसएस
carriers.
दी है
For
यहाँ पे।
example, if you are having dengue
ठीक है,
virus is carried
कलर
by
हमने
mosquitoes.
वाइट दिया,
uh,
उसकी जो
Chikungunya
डायरेक्शन है वो
virus,
हमने
which is
राइट
carried by
दी, ये पेंडिंग में
mosquitoes.
हमने
So there are different
रेटिनोसिस्टीन भी
carriers.
लगाई,
So
ये
cotton leaf
इसकी
curl virus, there
छोटी
is a
बारी के
carrier which is
काम
known as
आती है।
white fly.
और नेपाल में जो
White
इमेज
fly.
है, जो
So these
हमारा
are the its
लोगो
scientific
है,
name.
ये हमने
It is a
उसी
scientific
इमेज का
name first and foremost.
सीएसएस दिया है, जिसमें विड्थ और
Is
हमने
having the scientific name is
डिस्प्ले हमने
Panacea
ब्लॉक किया है, बॉर्डर हमने सॉलिड रखा है और रेडियस लगाया
Panacea
है,
turbacea.
जिससे वो सही हो जाए।
Panacea turbacea is its scientific name. And
ओके, उसके बाद
when it comes to
इसके बाद H1
white
जो
fly,
हमने हेडिंग दी है,
is
एजे
basically
कैट क्लब की, जो इधर हमने नाम लिखा हुआ है एजे टेक
sucks
क्लब,
the
उसका सीएसएस हमने जो किया है
cotton
वो
leaf
कलर वाइट और
plant,
विड्थ और
the
मार्जिन हमने
leaves of the
लगा दी।
cotton plant, as a
और
result
उसके
of
बाद
which
बॉडी का जो आपको याद होगा
it
हमने
causes to spread virus.
टैग लगाया था,
The cotton leaf plant,
ये उसके
the
बाद बॉडी के टैग में
it
हमने
causes
फिलहाल
infection.
खास किया नहीं है, क्योंकि
Zoophage. Zoophage includes
ये
these are
रहा
half
बॉडी का
full DNA,
टैग।
half full
बॉडी के टैग के
ionized.
अलावा हमने दूसरा सेक्शन लगाया था जिसमें हमने
Um,
एक्टिव वॉइस लिखा
these
था।
are
अब ये
viruses
बॉडी
which are
टैग
of different shape,
इधर देखो मैंने
different
खाली
genes.
छोड़ा है,
For
बॉडी
example,
टैग इसलिए
HIV.
खाली छोड़ा है, अगर मुझे पूरी बॉडी का कलर जो है
One more
वो
thing that
अगर मुझे
viruses are
ब्लैक
specific in
करना
nature,
हो
which
या
means a
वाइट
particular
करना हो,
virus
तब
infects
मैं
a
ये
particular
बॉडी
host.
टैग
For example,
इस्तेमाल
if there's
करूंगा।
tobacco
उसके
mosaic virus,
अलावा
tobacco mosaic
अब
virus
जितनी
is
भी
only
चीजें
into
मेरे पास
tobacco
इस्तेमाल
plant
करूंगा वो मैं
and the
सेक्शन लगाऊंगा,
plant
और
which are
एक्टिव
belonging to
वॉइस
the
जो
family
मैंने
of
आपकी
tobacco
लिस्ट
plants.
बनाई थी
For
उसका
example,
हमने ज्यादा
tobacco
कुछ
plants are
दिया नहीं,
belonging to
बस
the family
दैट्स
Solanaceae.
जो
These
थी
only
अलाइन वो
infect
सेंटर दिया था हमने। जैसे देखो ये
tobacco,
लाइन
only
सेंटर में आ रही है, एक्टिव
tobacco
वॉइस
plant
ऑफ
and
टैग
other
क्लब, ये जो
solanaceous
ब्लू
plant.
में आ रही है।
Likewise,
और मैंने इसकी
cotton leaf curl
हेडिंग लगाई
virus can
थी,
infect
मत और मैंने
cotton plant
बैकग्राउंड
and
थोड़ा
other
ब्लू किया
family
था।
plants such as
और जो
melanaceous
इसके अंदर
plants.
एल आई लिखे हुए थे वो उसको मैंने
Likewise,
चॉकलेट कलर
there
दिया
is
था, और लिस्ट स्टाइल जैसे मैंने आपने टेस्ट
HIV.
डेकोरेशन
HIV is a
किया था,
virus which can also
और
infect
लिस्ट
humans.
स्टाइल टेस्ट डेकोरेशन जो
And
लिखते
poliovirus.
हैं टेस्ट, वैसे
Poliovirus,
बात सेम ही है,
its
टेस्ट
actual
डेकोरेशन
name is
लगाओ या लिस्ट
coronavirus,
स्टाइल, वो
which causes
चीज एक ही है।
poliomyelitis.
तो इधर
And
मैंने
which
फिर चूंकि यूएल की मैंने
and
लिस्ट
which can
बनाई हुई थी,
causes
इसलिए मैंने इधर लिस्ट स्टाइल रन कर दिया, जिससे
poliovirus,
इसके जो
poliomyelitis,
डॉट्स थे
and that
वो
basically
गायब हो
is
गए। ठीक
common
है,
and it can
ओवरफ्लो भी आप कर
infect
सकते
humans
हो,
and
वैसे ये
primates.
आपकी
Primates
डिपेंड
are
करता है।
those
और चीफ की
animals
जो मैंने
which are
पिक्चर
having
लगाई थी
flexibly
जुबैर की,
tamed
जैसे नीचे
tails.
देख रहे हो, फिर इसका मैंने विड्थ
And
और
one
रेडियस
more
के
thing over
बारे में
here,
समझाया था, इसकी पूरी
uh,
विड्थ और रेडियस। अब
HIV is there,
जब से
polio
क्लासेस
is
हमारी
there,
शुरू हुई हैं
and
यहाँ तक,
one more thing,
कोई भी अगर बात समझ ना आई हो,
hepatitis.
कल तक मुझसे
Hepatitis
पूछ लें, कल हम
sulfur.
आगे चलेंगे। अब क्लब की मेरे ख्याल
HIV
में
is
कल आपका
circular,
क्लब ऑन होगा,
polio is
और
finger
कल
flies,
आपको
and then
एक्टिव हो
hepatitis
जाना है
B.
टाइम पे, और मैं भी कोशिश करूंगा थोड़ा जल्दी आ जाऊं।
Hepatitis B
तो
is
कल से फिर
also
हम आगे शुरू करेंगे। और यहाँ तक
hepatitis
किसी
B
को
is
कोई भी
having a different
कंफ्यूजन
type.
है तो बराए मेहरबानी मुझे मैसेज करें। कई बार मैसेज देखने में,
Now,
रिप्लाई देने में
hepatitis B
देर
is
हो
also
जाती
known as
है,
serum
तो
hepatitis.
एक
We are
कॉल
going to
कर दिया
discuss
करें
about
जिससे
it
मुझे
in
पता
detail
चल
in
जाए
our
किसको
upcoming
क्या
class.
काम है। ठीक है,
Moreover, when
अल्लाह
it comes
हाफिज।
to
अभी
some
आप
other
दूसरे
parts,
विकेट में।
bacteriophages. Bacteriophages are also having different types. Bacteriophages are also having different types, which includes T phage, T phages. Now these are also divided into different types: T4 phages, T1 phages, T6 phages, which we are going to discuss in the life cycle of bacteriophage. X phage. One economical importance of these bacteriophages is that whenever we are having savage water, in order to clean the savage water, uh, there is the utilization of bacteriophages, such that when bacteriophages are released into the savage water, they tend to eat up the bacteria. They tend to kill the bacteria, due to which the water is then cleaned up from bacteria. As well as there are other importance as well, that if water, usually we see that uh, there are different actually cases for each and every virus. There, the importance of each virus is there as well. In some cases, those viruses which can kill bacteria are also beneficial to humans. One more thing over here, that here viruses which enter the cell, enter the host cell. Viruses enter the host cell. When viruses enter the host cell, they tend to create the multiple copies utilizing the metabolic machinery of the host cell. And in that process, they, as they create multiple copies, these multiple copies causes to increase the mass of the cell, increase the mass of cell. As it increases the mass of cell, it causes the specific gravity of the cell to be increased. One more thing, that here non-living characteristic of virus is that they are having high specific gravity, but living organisms are having low specific gravity. So here, as they increase the mass, increase the specific gravity that causes to increase the capab. As they increase the mass, increase the specific gravity, it causes to either burst the cell, or in some cases the viruses produced are turned out outside through exocytosis. Next phages, phycophages. Yes, phycophages. Phycophages are those which attack algae, mycophages are those which attack fungi. Now coming to the major classification we are going to study today. Basically, there was a virologist named as uh, Baltimore. David Baltimore was a virologist, a Nobel Prize winning biologist, Nobel Prize winning virologist in 1971. He classified viruses into seven classes. Three major groups were there, and he classified each of them into seven major classes. Let us discuss that in detail here. As you can see, the genetic on the basis of the genetic material genome, as well as on the basis of their mode of replication, it was classified into seven major classes. Now on the basis of seven major classes it was done. After that, in recently in 2018 to 2019, the classification of Baltimore was further modified, and then it was modified on the basis of its ancestral evolution. When it comes to the groups, these are basically the classes. Now there are two classes. First includes the double stranded DNA and single stranded DNA. These are the two classes which are classified into the single group, which is the DNA virus. And here we are having these classes, which includes double stranded RNA, positive single stranded RNA, plus plus stranded RNA virus, minus stranded RNA virus, which are classified into RNA viruses. On the other side, these are the classes classified into groups known as RNA viruses, and these are the retroviruses. Retroviruses, RT viruses, retroviruses, or reverse transcribing viruses, or we can also call them as um, reverse transcribing viruses, RT viruses, or retroviruses. Now, these retroviruses are basically hmm, having characteristic that here they are having enzymes reverse transcriptase, due to which they perform reverse transcription. Now, coming to this point, that here first let us discuss about the double-stranded DNA. Double-stranded DNA viruses work in such a manner that double-stranded DNA viruses are basically having a double-stranded DNA which they utilize as a genome. When they enter inside the host cell, the double-stranded DNA basically causes to perform transcription inside the nucleus. The DNA viruses perform their function in nucleus as well as cytoplasm. RNA viruses perform their function in cytoplasm only, while retroviruses perform their function in nucleus as well as cytoplasm. So these are some key points. DNA viruses basically are having two characteristics. DNA viruses perform first transcription using the cell's metabolic machinery, and then translation using the cell's metabolic machinery. Once they perform transcription, they produce mRNA. As they produce mRNA, this mRNA either it is utilized for the process. mRNA is produced, which is either utilized for the process of protein synthesis, or it is utilized through another process to form DNA. Now here, protein which is synthesized utilized to form its multiple copies. Moreover, examples of these double-stranded DNA viruses. One more thing, double-stranded DNA viruses are not found in plants. These are not phytophages. These are zoophages, bacteriophages, but these are not phytophages. These are those viruses which cause uh, cancer in animals. They cause cancer in animals. Um, Examples of these is Rio virus, double-stranded DNA virus, Rio virus. Second classification is single-stranded DNA virus. Single-stranded DNA viruses are those viruses which are, one more thing, there's a mistake over here. It's not Rio virus. Instead of Rio virus, we can consider another example of double-stranded DNA virus. Another example of double-stranded DNA virus includes smallpox virus. We can consider it. Smallpox virus is a double-stranded DNA virus. One more thing over here, that here we are also having another characteristic, positive DNA virus. Positive DNA virus is having basically uh, positive DNA virus. Basically, it is a single-stranded DNA virus. It is a DNA virus which is having a single strand which runs from 5 prime to 3 prime. Why it runs from 5 prime to 3 prime? Because the strand which it replicates inside the host cell would be 3 prime to 5 prime, and we know that during replication of DNA, the first strand which is formed is the 5 prime to 3 prime strand, which further replicates its complementary strand known as 3 prime to 5 prime strand. So when it goes inside the host cell, it forms 3 prime to 5 prime strand and forms a double stranded DNA, as you can see in the diagram. And after that, the double stranded DNA undergoes transcription and then forms translation. Basically, the double stranded DNA here formed is either utilized as genome, either it is utilized as the genome, or it is utilized for the protein synthesis by performing transcription and translation. Example of single-stranded DNA virus includes parvovirus. Parvovirus is the fifth disease in young children aged 1 to 6, which includes diarrhea, which includes a symptom, a disease in which there is diarrhea, vomiting, respiratory tract infection, as well as diarrhea, vomiting, respiratory tract infection, fever. So this is what we include in single-stranded DNA virus. One more thing over here, that as you can see in the group 1 double-stranded DNA viruses, double-stranded DNA virus also include example of rhinovirus. One more thing here, in the other example we are having double-stranded RNA virus. Double-stranded RNA virus includes the example of reovirus. Now double-stranded RNA virus are having characteristic that they basically function in cytoplasm. So inside function, they perform function in cytoplasm and utilize an enzyme inside the cytoplasm known as RNA dependent RNA polymerase, due to which they they're utilizing their double-stranded DNA, they double-stranded RNA, sorry, is utilizing their double-stranded RNA, they form a single-stranded mRNA, which basically they form single-stranded mRNA. This process is known as transcription, and from that transcription formed mRNA undergoes translation and forms protein synthesis. Now mRNA which is formed over here is either utilized for protein synthesis or for genome formation. Example of this includes reovirus. After that, there are two classifications of single-stranded RNA virus, which includes the class plus sense single-stranded RNA virus and minus sense single-stranded RNA virus. Now, positive sense single-stranded RNA virus are those viruses which are having a single-stranded RNA, which is basically 5 prime to 3 prime, 5 prime to 3 prime, which means these are having a positive feature, that once they are having 5 prime to 3 prime RNA, this resembles the RNA of the messenger RNA, which is 5 prime to 3 prime as well. Messenger RNA is 5 prime to 3 prime, so they do not need to undergo transcription. They directly undergo translation without transcription, so no transcription occurs in pillar stranded RNA viruses. Examples of these include a good example of this is hepatitis C, COVID-19, as well as hepatitis C, COVID-19, dengue virus. All of these are plus stranded RNA viruses, and minus stranded RNA viruses includes examples of minus stranded RNA viruses includes, first and foremost, these are known as minus stranded because these are from 3 prime to 5 prime, which means these are inverse of the R primer of mRNA, due to which these are 3 prime to 5 prime, as a result of which they need to undergo transcription through the enzyme RNA dependent RNA polymerase, as a result of which we can say that as they require RNA dependent RNA polymerase to convert 3 prime to 5 prime to 3 prime. This process is known as transcription, and from the transcription, once mRNA is formed, that mRNA is utilized for the formation. mRNA is utilized for the formation of proteins through translation. After that, this includes example of rhabdovirus and paramyxovirus. Paramyxovirus causes measles and mumps, Rhabdovirus causes rabies. After that, when it comes to some other sort of viruses, uh, there are some other sort of viruses such as the retroviruses. Retroviruses characteristics are already defined. Basically, group 6 and group 7. And um, these are include this includes RNA virus, single-stranded RNA with DNA intermediate viruses. How do they work? These are basically having a single-stranded RNA, two molecules of single-stranded RNA, two identical molecules of single-stranded RNA are present. And how do they work? Actually, they actually utilize a single-stranded RNA to perform reverse transcription. As they perform reverse transcription, they produce a double-stranded DNA using the enzyme reverse transcriptase. As a result of which, this double-stranded DNA is produced, and after the double-stranded DNA is produced, it utilizes another enzyme which is present within its own structure known as integrase. Integrase and reverse transcriptase are its own enzyme which perform the two events, reverse transcription and integration, which are performed by its own enzymes of metabolic machinery. So here, reverse transcriptase first and integrase integration is performed by integrase, as a result of which the DNA of host cell is integrated with the DNA of uh, the virus, which is the RNA single-stranded RNA virus with DNA intermediate, due to which double-stranded DNA is integrated. After that undergoes transcription and translation, as a result of which now the double-stranded DNA which is produced over here, this double-stranded DNA causes production of mRNA through transcription, and that mRNA which is produced over here, now it has two options: either it is utilized for protein synthesis or translation, or it is utilized for genome synthesis. It can be directly utilized for genome synthesis. uh, it can be utilized as genome because mRNA as well as the single-stranded RNA are from 5 prime to 3 prime. Apart from that, uh, when you can see uh, examples of this is HIV. After that, um, second class is DNA virus with RNA intermediate. DNA virus with RNA intermediate are double-stranded viruses, and these double-stranded viruses are basically having characteristic that uh, these double-stranded DNA viruses, first and foremost, they utilize. These are having double-stranded DNA with RNA intermediate. How? Let's see. By the way, there is only single molecule of double-stranded DNA. Now, that double-stranded DNA is structurized in such a manner that here, for suppose if there is a double-stranded DNA present within their structure, this double-stranded DNA. One more thing I wanted to mention over here, that as they are having double-stranded DNA, this is not complete double-stranded. They have double-stranded DNA, but at some intervals they are also having a single-strand DNA, which means, for suppose if there is a double-stranded DNA, this is a double-stranded DNA going on, but mm, at some intervals these are having a single-stranded DNA. For suppose if this is a double-stranded DNA, at some interval it is having a single-stranded DNA. In that manner, these are having their DNA, and basically first and foremost they convert their DNA, double-stranded DNA, into RNA, into the RNA. From RNA they undergo reverse transcription, due to which they produce DNA, and with by the help of the double-stranded DNA they undergo integration through integrase enzyme, due to which they integrate their DNA with the host cell's DNA, due to which after that they perform transcription, as a result of which they form. Now here, when double-stranded DNA is integrated, that double-stranded DNA has two options. Either it forms protein synthesis under translation and transcription, or it undergoes, or it is utilized as a genome for the virus, which is going to be its progeny. So as a result of this, it's either utilized for genome synthesis or protein synthesis. Now, for protein synthesis, transcription, translation occurs in protein is synthesized. Example of these includes hepatitis B. This is a brief introduction, and hopefully you enjoyed this class. Hopefully, we'll meet again in another one.